you should be unshakable in the 3+3 design, like a 10-year-old when you ask her, what is 3+3? And today we will get you there. Welcome, everybody. You have clicked in a series of teaching videos explaining clinical development in oncology. This time, we will explain the 3+3 design for those who execute clinical protocols and train it into your subconsciousness so you will never need to think or read about it again. Then we will have some tips for those of you who will use the designs when writing the protocols, and finally some thoughts for those of you who will want to develop our designs further. My name is Johannes Wolf. This is recorded in May 2024. Oncology is the medical field in which we treat cancer. Drug development is a field within the pharmaceutical industry in which drugs are brought from discovery to clinical approval. This is typically done in 3 phases. Very much simplified, phase 1 is the dose finding and the endpoint is toxicity. We are today talking about 3+3 design, a very commonly used study design for phase 1 studies. Phase 2 studies have 20 to 40 patients. They look at preliminary efficacy, and the typical endpoint is response. Phase 3 studies have hundreds of patients, compare the new treatment to standard of care, and the typical endpoints are time to event endpoints such as overall survival. Here you see a study design schema for a phase 1 study and preliminary results of it. You see the patients received 25 milligrams in the beginning, and the final patients were moved up to 350 milligrams. In the first cohort of 25 milligrams, 3 patients were treated and no dose-limiting toxicity was observed. The same happened when the dose was escalated until 250 milligrams were reached. There, 1 patient experienced a dose-limiting toxicity, and therefore the number of patients was expanded from 3 to 6, 3+3 design, but no further dose-limiting toxicity occurred. The dose was further escalated. The same happened in the next dose, but 1 dose level higher at 350 milligrams. Both of the first patients that were enrolled experienced dose-limiting toxicities, and therefore the study escalation was not carried further. There were responses, tumor shrinkages observed. And you see the number of tumor shrinkages went up at the time with the dose being escalated. But 3+3 design only looks at the number of patients and the number of dose-limiting toxicities. It is a rule-based system, and we will explain it next in 4 different ways. 3+3 design protocols typically describe the design in words and in graphics. This is how the words can look like. It describes that you should start every dose level with 3 patients. If there is no dose-limiting toxicity among the first 3 patients of a cohort, then the recommendation is to escalate to the next higher dose level. If there is 1 dose-limiting toxicity among the first 3, then 3 additional patients will be added to the same dose level, 3+3. If there are 2 or more dose-limiting toxicities among the first 3 patients, then that dose level is too high and de-escalation to the previous dose level is recommended. This goes for a while until the maximal tolerated dose can be determined. The maximal tolerated dose is a dose level where 6 patients have been treated and the total number of patients with dose-limiting toxicities is 0 or 1, but not 2 or more. And the next higher dose level has been tested and found too high. Most protocols describe 3+3 design not only in words, but also in a schematic. This is a typical schematic how it is described. You enter 3 patients in a dose level. If there is no dose-limiting toxicity observed, the next step is escalation to the next higher dose level. If there is 1 dose-limiting toxicity among 3 patients observed, then 3 more patients are entered at the same dose level, 3 plus 3, ending with 6. Once 6 patients in the same dose level are enrolled and 1 or no dose-limiting toxicity was observed, then you may escalate to the next higher dose level again. If there are, however, more than 1, so 2 or more patients observed with dose-limiting toxicities, then that dose level is considered too high, and the previous dose level will be considered the maximal tolerated dose. If when entering 3 patients into a dose level, 2 or more patients were observed with dose-limiting toxicity, then that dose level is considered too high, and the previous dose level is the maximal tolerated dose. That schematic is fairly easy, understandable, similar to the way how the words described it before. But both the words and the schematic in reality leave decisions open. For example, at this point, it is not true that there is always a stop in the study. That depends on how many patients had been enrolled in the previous dose level. If the previous dose level went this way, 3 patients, no DLT, escalate, then there were only 3, and you cannot determine an MTD with only 3 patients. You need 6. Remember the last sentence of the word type description. So when this happens, you need to de-escalate, add 3 more patients to the previous dose level so that it will eventually be 6. The same is true here, and there are other problems, other decision points that are not reflected in the schema either. For instance, when the entering 3 patients and you find no dose-limiting toxicity and it tells you to escalate, but there is no dose further there to escalate. This was the highest dose that the protocol had given. You have to make other decisions. Repetition is the mother of memorization. You have seen it now in 2 different ways. We will now play a 3rd way. These decisions are typically made in committees. These committees have various names. I know a company that calls them dose escalation meetings. Another company calls them SRC, Safety Review Committee, or CRC, Clinical Review Committee. Think yourself as being the chair of that committee. You now have 3 patient data to evaluate. There are 4 possibilities. There could be no DLT observed. 1 of the 3 patients could have a DLT. 2 or 3 of the patients could have experienced DLTs. In general, 3 plus 3 design, this thinks this dose is probably too low. We should escalate. This dose might be right. We should add 3 more patients. And this dose is too high. We should de-escalate to the next lower dose. But it is a bit more complicated. So here, when the dose is probably too low, it depends what the next higher level has to offer. If the next higher dose level is actually available, then yes, by all means, you escalate and the next 3 patients will be enrolled in the next higher level. But what if the next higher level doesn't exist? This is already the highest level that protocol has described, or the next higher level has already been tested and found to be too high, too toxic. Yet you only have 3 patients here. Then you add 3 patients to make it 6. Now, if there is 1 in 3 observed, then there is no complicated decision. You add 3 more to the same dose level. If the dose was too high, then the next action depends on what we know about the lower dose. If the next lower dose has been tested with 6 patients and those had none or 1 DLT, then the study has succeeded. You know the maximal tolerated dose. It is the previous dose level. This dose level was not tolerated. The maximal tolerated is the previous. And that is a reason to celebrate. If the previous dose level, however, had only 3 patients, and among those 3 patients there was no or 1 DLT observed, then you de-escalate. Now, of note, 1 observed DLT should not occur, because if this was among those 3 patients, they should have gone down that way, and there should be more than 3 patients by now. But medicine is complicated. Clinical trials don't always go the way you want it. If that happened, you may have to look in protocol deviations and maybe file a few papers to get it right. But the 3+3 design still allows you to make this decision in this meeting. And the decision will be de-escalate. Add 3 more patients to the lower dose level so that you come from 3 to 6 and that you then can declare the MTD. However, what if this level was the lowest, the first? There is no lower level in the protocol described. That's really bad luck, because then you have to stop the trial. You cannot declare an MTD. This is basically a trial failure. The doses described in the protocol were too high. Congratulations! You have survived your first SRC meeting as chair, and now you're heading towards the second. This time, there will be 6 patient data to be evaluated and decisions to be made. Fortunately, it actually is a little bit easier because there are only 2 possible decisions, not 3 like there was before. Either the dose is considered tolerable by a 3+3 definition, or not tolerable. Tolerable would be if there was no patients that experienced DLTs or 1 patient who experienced DLTs or 1 DLT. It will be considered not tolerable if the number of patients experiencing DLT is 2 or higher. But it is more complicated. For the first group, the good group, it depends on what the next higher level has to offer, what data are available there. Assume the next higher level is open. It is available, it hasn't been tested, then you indeed escalate. You go up. Now, just one word of caution. If there were actually 0 in 6, then that situation shouldn't happen, and we have a protocol deviation again that will cost some paperwork, but it will not affect your ability to make that decision. You can still escalate and make this all move forward. What if the dose level you're looking at today is the highest dose level, and the protocol doesn't have any further dose levels defined? A trial stops. You cannot declare a maximal tolerated dose, because for that, the next higher dose level would have needed to be explored and found to be not tolerable. The only thing you can declare is a maximal administered dose, an MAD. And what if the next higher dose level has been tested and was found to be not tolerable? Then you can celebrate because your trial has succeeded. You can declare a maximal tolerated dose. It is this dose level, the dose level you just tested. Go and celebrate. Let's look at the other side. The side where the finding indicates that the dose level is not tolerated, is too high, is toxic. Then it depends on the previous dose level and the lower dose level. If the lower dose level has already been tested and there were 6 patients enrolled and those 6 patients had 0 or 1 DLT, then you can again celebrate. but this time it's not this dose level that is the MTD, it is the previous dose level. The previous dose level is the maximal tolerated dose because this dose was not tolerated. Success. What if the previous dose level, the lower dose levels, had only 3 patients enrolled? Then you cannot declare the MTD because you need 6 patients to declare the MTD. You need to de-escalate, add 3 more patients to the lower dose level, and then have another meeting. What if the dose level that you're looking at, those 6 patients dose level, actually was the lowest dose level in the protocol? The protocol doesn't have a lower dose level defined. Then you're in the same situation as you could have been a month ago with the 3 patients. This trial is a failure. The doses that were defined for the trial were too high. You have to go back to the drawing board. Okay, now you know 3+3 design. There is nothing more to learn. There is just to be practiced. So it goes in subconsciousness and you can use your thought processes for more complicated things. That's what we're going to do right now. I'm going to ask questions. You're going to answer. You're going to answer and speak it loud. That helps. So here is the first situation. 3+3 design, the first 3 patients have been enrolled. None of the 3 patients has a DLT. What do we do? Please answer. Correct. You go up with the dose. You enroll 3 patients in the next dose level. So here's what happened. Among the next 3 patients, 2 were without DLT, but 1 patient did have a DLT. What is your decision? What is to be done next? Speak it out loud. Correct. You add 3 more patients, 3 plus 3. Now you have 6 patients, 1 of which had a DLT. What is your next decision? Correct. 6, 1 DLT, next higher level not determined means go up. You did go up. Here are the data of the next 3 patients. 1 was fine, no toxicity, but 2 had DLTs. What is your next decision? Correct. You're done. You can declare the maximal tolerated dose. This dose was not tolerated. This is the maximal tolerated dose. You have 6 patients treated. Only 1 has a DLT. 3+3 design, done. Congratulations. Next test. First 3 patients enrolled, you moved up. Next 3 patients enrolled on the next level. What do? Not really hard. You go up again. Here are your next data. Next 3 patients, 2 patients had DLTs, 1 was fine. What is your decision? Speak it out loud. Okay, you de-escalate because this dose level was not tolerated, but the lower dose level didn't have the necessary 6 patients yet to be declared an MTD. Now, here's your next situation. You now have enrolled the 3 further patients. 1 of those had a DLT, the other 2 did not. What is the next step? Correct, you're done. You have declared, you can declare the MTD, the maximal tolerated dose. The higher dose level was not tolerated. This dose level is tolerated by the definition of 3+3 design because only 1 out of 6 patients had a DLT. Next test. Here we go in the next situation. You had the first 3 patients enrolled. There was nothing, you escalated. In the next dose level, however, only 2 patients are enrolled and you're getting a call. Your ClinOps colleague warns you there will be a DLT registered in the EDC tonight. The patient had a DLT. What do? Well, this is a bit more complicated because there is no SRC meeting scheduled for that time. You have to do some calls. But obviously, if 2 patients had DLTs, this dose level is not tolerated, and you should not allow a 3rd patient to be enrolled on that dose level. Instead, call an ad hoc SRC, open the previous dose level again, and enroll 3 more patients on the previous dose level. Here are your data. You have enrolled those 3 patients. No DLT, DLT, no DLT. What is your next decision? You're done. You can declare the MTD. This dose level is the maximal tolerated dose. The higher dose level was not tolerated. You could determine this after only 2 patients being enrolled. Next situation. You have enrolled 3 patients on the first dose level and there was no DLT. You escalated and you escalated again and you escalated again. And now you have reached the maximum planned dose in the protocol. There is no higher dose level allowed in the protocol. What do? You enroll 3 more patients. Now here are your data. You have now enrolled 6 patients on the maximum planned dose, and there is no DLT. What do we decide? Well, you can't go up because there is no further dose level in the protocol ready for you. You can't go down because the lower level dose levels have already been cleared. There is no toxicity, and that's confirmed with those 6 patients on the highest dose level. You have to stop the study. You cannot declare a maximal tolerated dose because this dose was tolerated, but you don't know if it was the maximum. The upper dose level that was not tolerated is missing to make such decision. What typically happens is a thorough data analysis. There are always data missing on PK and PD, things where a lab value is lagging behind the safety information a little bit. You pull all that in, you review the safety profiles, you make sure that all data are source verified, everything is clear, and you also look at the efficacy data. All that together will allow a decision what to do to be done next. Next example, 3+3 design, only 1 patient enrolled. This patient has a DLT. What do? That's a tricky one. And it's a trick question because 3+3 design actually does not tell you what to do. This situation goes well beyond what the decision algorithm of 3+3 can possibly give you. It could be that the entire program needs to be closed. For instance, if this first patient was a patient with a healthy heart, and that was well documented in ECG, echo, and history and family history, and had no further risk factors for anything, yet he developed a massive heart attack shortly after the infusion had been completed, then was resuscitated, went to the ICU, and could not be rescued. But causality is out of the question. It came from the drug, and it was deadly. The patient passed away, grade 5 adverse event. That would typically be the end of the drug development of this drug. On the contrary, maybe this first patient should actually not have ever gone on the protocol because that patient was allergic against one of the substances that are in the drug, and there was an infusion reaction. And the DLT criteria are written a little sloppy, and they kind of wrote that grade 2 infusion reactions are DLTs. Yet that was really minor. They gave a little dexamethasone and everything was gone. The patient could go home 4 hours after everything has happened. And in retrospect, they found out the patient was allergic to a known substance of it. The patient should never have been enrolled. It's a protocol deviation. Nonetheless, that is a DLT and will always count as a DLT. But and then let's make that example even clearer. A week after the infusion, the principal investigator gives you a call. This patient had relapse recurrent acute myeloid leukemia, and the leukemia is gone. After only 1 infusion, the leukemia is gone, no more blasts in peripheral blood. You wouldn't kill that drug. You would carry on. So this decision is not a 3+3 decision. This is a decision that needs to be made by other sources of information. Let's assume you made that decision, and the decision was to carry on. But really cautiously, After each patient, you put in now a safety evaluation, and here is your data. This didn't happen, you didn't stop, you carried on. The next patient had no problems. No problems, no DLT, nothing else. What do you do? Wow, that's an easy one. Yeah, no, it was an easy one. You enrolled the third patient of that cohort. Oh, that third patient also has a DLT. Now we have, among 3 patients, 2 patients with DLT. What do we decide? Correct. 3+3 design stops the trial. This dose is not tolerated. You cannot go higher. You cannot go lower because it was the first one and there is no dose level -1. Some protocols have a dose level -1, but not in this example. So in this example, it's the end of it. You need to look at all the data, rethink, possibly amend the protocol, possibly write a new protocol. But in any case, this one is stopped and it is kind of a trial failure. That was it. Congratulations, you made it. All 3+3 knowledge is now in your head. We are moving on. Promotion. You're now not following a protocol. You are now writing a protocol. Here are a few tips when you have to write a protocol and you have made a decision that 3+3 design will be the design of this study. Number 1, starting dose. There is no way you can write a 3+3 design unless you have a definition and a decision about the starting dose. That is a complex process that may include animal data, probably the most important data. There may be a receptor occupation data that are not even in cells, that are biochemical data. There is PK modeling, PK/PD modeling, and there may be human data from healthy volunteers or human data from other phase 1 studies. All that together helps you find the right dose. A common approach is 1/6 of the highest non-severely toxic dose in non-rodents. All that process is done by various professionals, and the clinical development specialist is only a minor part of that decision process. But I'm recommending for the clinical development person to look at other protocols of similar drugs that had been successful in the past and validate what comes out of the starting dose discussion, and also the dose levels thereafter and the maximum dose on the protocol. There is another very big discussion around the definition of DLTs, dose-limiting toxicities, and part of that is the definition of which adverse event will count as a DLT. This includes causality. Obviously, when we want to find the right dose and we define that dose by the toxicity caused by the drug, we don't want to be led in the wrong direction by adverse events that don't come from the drug, come from the primary disease or from something else. So it is well advised to have a paragraph regarding causality, making sure only those severe adverse events that count, that were caused by the drug, are dose-limiting toxicity, not only severe adverse events, but adverse events. Secondly, the second part is indeed the severity. Commonly, CTCAE grade 3 or higher is used as primary definition for the severity, and then exceptions from that are defined underneath. Among the exceptions may be nausea, vomiting for 1 day only. Often SAE, severe adverse events, are used in the definition, and very commonly when the dose was changed or the dose was interrupted, the patient was taken off the study because the investigator had the opinion that the drug caused toxicity. That is often also a part of the definition of DLTs. There are more. I would say look at other protocols and copy some of it, but think about it before you copy it. Almost equally important is the DLT period. It is easier to define. Commonly, the first cycle of treatment, and that's often 21 or 28 days, is used for that. Sometimes, in particular by drugs that have long-lasting, slow-occurring adverse events, more than one cycle. That obviously makes the protocol much slower. And then there is the DLT population. Who counts as being a patient among the three of a 3+3 design? Obviously, any patient who has experienced a DLT within the DLT period will have to be counted, regardless how much drug that patient got. But what about the others, those that were treated but did not experience dose-limiting toxicity? Well, if they had all of the drug, then they count. But what if they only got some of the drug? What about a patient that was treated for 1 week and then was tired of the hospital, got in some kind of fight with some kind of a nurse and goes somewhere else? That patient has received the drug. That patient will be part of the safety population when the study is eventually described. But will that patient be part of the DLT population? No, because before we count somebody as not having had a DLT, that patient needs to have a decent amount of the drug so that the data can be believed. And the typical definition is 80%. You can use other definitions, but don't forget to define it. The last recommendation is to be really cautious with the definition of the committee that makes these decisions. This may not all be in the protocol. Some of this may be defined in a committee charter. Among the recommendations I have there is make sure the committee has permission to override the 3+3 rules. 3+3 is, after all, a very primitive design, and it's rigid. The only flexibility comes through the definition of the DLTs, but that is sometimes not enough. Sometimes clinical decisions need to be made, and the committee needs to have the power to be allowed to do those things. Second recommendation, be careful how to define who participates and who has the right to vote. Obviously, the medical monitor from the sponsor should be one of those. That's typically the chair of the committee. Sometimes there is a CRO that also has a medical monitor, and that person may be a part of the committee as well. There is the statistician, clinical scientist, the safety physician. All of those, or some of those, can be part of the committee, and it is important to define who should be there. Also important, and maybe the most important thing, is how is the decision made? This could be a quorum of, let's say, 75% of the participants have to be there, and then the majority of those makes the decision if the dose gets escalated, de-escalated, or if the dose stays where it is. Then there is consensus. I have seen charters that require consensus, meaning everybody has to agree. And to make matters worse, some of those charters also require that all of the possible participants have to be there in that meeting. Don't do that. That is almost a certain hangout for the protocol. There will be always the risk that somebody can't make it to the meeting. Some principal investigators are not part of the study anymore. They don't even need to come. And you have them in the charter as a necessary member, and you have to reach consensus. Don't do that. Find a different way. I have seen decisions in charters or workflows or SOPs where simply the chair makes the decision. Everybody has to be heard. All the data need to be in. And then the chair makes the decision. We move up, we move down, we stay. Also possible, very practical. Obviously, people have some concerns about that. But the worst of all is not to describe it. Then you are heading towards trouble and a hang-up of the protocol. There are further developments beyond the classical 3+3 design. That includes rule-based models, derivatives of the classical design. The accelerated version uses smaller cohorts than 3. You can go with a single patient cohort. That is done when the starting dose is real uncertain. People want to be really, really cautious and go really low with the first dose, but then they don't want to treat too many patients with those likely underdosed doses. Then you can do one patient cohorts, and the acceleration from one dose to the escalation from one cohort to the next cohort may not depend on DLTs, but on much, much less adverse events. Typical thing is one patient enrolled. If that patient has neither efficacy nor any adverse events, grade 2 or higher, then you move up to the next level with only one patient. If any patient has developed a grade 2 adverse event, then it moves from one patient cohorts to 3 patient cohorts, and then it is a classical 3+3 design. That is to have less patients enrolled in the lower dose levels. The second derivative is the opposite, having more patients. They're called the so-called backfills. That means the 3+3 design runs normal as typical with DLT definitions and dose escalations, but then once the MTD is decided, you allow further patients to enroll in the dose levels that have already cleared. Now, I have personally somewhat ethical problems because we know those doses are too low. These patients have cancer, and you give a drug that you know you could give at a higher dose. I mean, you have to at least think about this. And then there is an operational practical issue with those backfills. When you put this in your protocols, make sure you think about what to do when one of those patients develops a DLT. A very practical and simple way to say this doesn't count. So DLTs and counting and 3+3 rules only apply for the classical dose escalation. The backfills play no role. That solves the problem. It's questionable if that's a very good way of doing it, but at least it solves it. And then finally, the problem with the first dose level being too high and in fact the drug should be dosed even lower than that can be solved with dose level -1 or -2. So those are dose levels lower than the first dose level you enroll. And that is another derivative of the 3+3 design. I think there's absolutely no problem, and that should be done much more often than it is done. 3+3 design doesn't need a lot of thinking. You can put it in subcortical thinking and just make it instincts. There are far more intelligent drug designs. Many of those are based on the so-called Bayesian mathematics. That is a different way of calculating probabilities. It includes information from the past, adds the current information, and then turns into probabilities for the future. An approach that requires more mathematical, more computer power, and a little different mathematics, but it is taking off. It's becoming very modern. Many things are now calculated with Bayesian, and that includes dose escalations. The CRM, continuous reassessment model, allows you to enroll more patients than 3+3 design. It allows you to go up and down and up again between dose levels, which, as you know, 3+3 design won't allow you. The Bayesian optimal interval design even allows you to have different dose levels in between not previously designed dose levels. Now, those complicated and intelligent things can be made simpler with software packages where you type in the numbers of patients and which patient had what, and then it tells you what the next patient should get. But it has some drawbacks, one of which is it's more complicated. If you have to explain why you're going up or down, the 3+3 design is a very simple thing that's very simple explained. Try to explain a Bayesian model and why the dose goes up or not down, or why you stay at the same dose level. That is difficult, and sometimes you need to explain things. Second, I've heard more than one good, experienced clinician say, I don't trust computer-based programs enough to let them decide what my patient gets as dose. I want to have a say in it. And then finally, a 3+3 design you can write if you're not a statistician. Those things I would strongly advise against attempting to do this without a full professional statistician. And to have that person, that statistician, with you when the study happens. Keep in mind, some of these things happen on the weekend. You need to have a good statistician if you do those models. With all that, they may be more intelligent, they may be faster. But 3+3 design still is very solid, and a good study design probably still the majority. More recently, there are compromises. There are model-assisted rule-based designs. They basically have a table very similar to 3+3 tables in the protocol, which tell you with how many patients, how many DLTs you go up, you stay, or you go down, or you eliminate a dose level. Commonly used among those is the modified toxicity probability interval design, MTPI. And then there are many more. So look at them, talk to other people before you make a decision. But 3+3 design is still, even today, a solid way of going. Those are study designs we already have. But the future will bring more. And I hope among the listeners are a few of those intelligent people that will make our future better and come up with even better study designs. Let me give you a few thoughts where I think we are lagging behind what we could do. First of that is the general concept that there is such a thing as a right dose for everybody. Look at these two people. It appears that they both have some joint problems, some orthopedic things, and maybe drugs could help them. Would you think that person should get the same dose as that person? Of course not. This, we give everybody the same dose, is particularly off with adult patients and oral medicine. Think about this. When we treat animals, a dog, an elephant, a mouse, we treat them per weight. Milligram per kilogram is the typical dosing way. When we treat children, we treat them per body weight, per body surface area. We include the age in the complicated algorithms, what dose that person should get. When we treat an adult patient with intravenous medication on the floor, we typically calculate that per kilogram body weight or per meter square body surface area. But when we treat an adult patient in the outpatient setting and we give them a tablet, then all of a sudden everybody gets the same dose. One tablet for everybody. Can that possibly be right? No, it isn't. We need to diversify doses for people. The different people need different doses. And the body size is certainly not the only thing and probably not even the most important thing. There are the dependencies on certain organ functions, liver and kidney function, on age that relates to those organ functions, and on metabolic characteristics. Our colleagues in Japan typically do not allow to give just the same dose that was tested in European or American wide populations. And they have very good reason. There is a metabolic thing that is different somewhat depending on the genetic background. And there are good stories, very exciting stories around it. Our colleagues in Japan have figured that out. Not everybody needs the same dose. There is an influence of genetic background. I would hope that in the future, we will have an average or standard maximal tolerated dose from which we calculate the derivations based on organ function, age, body size, and metabolic characteristics. Obviously, our phase 1 studies then need to take care for that as well. They could even now. A patient that gets a dose-limiting toxicity that was half the size of the others, that may mean something different than a patient that had the same size and got the dose-limiting toxicity. Supportive care. can change toxicity. Supportive care, for instance, for nausea, vomiting, a common adverse event in chemotherapy in drug development in oncology. We have fantastic drugs, very, very effective to prevent or treat vomiting, nausea. If you give a drug causes nausea, and you develop a dose-limiting toxicity based maximal tolerated dose based on the nausea, but in reality, you could go 3 times higher if you just gave a tablet against nausea. What does that NTD mean? It means nothing. You have to include at least standard supportive care to find out which dose is really tolerated. The initial dose may be calculated with a maximal tolerated dose and sort of an average dose to be given. But after the initial dose, there is a wealth of far more meaningful information, and that is the tolerability of the individual patient. It is good medicine if somebody develops adverse events in response to the first drug dose that you give the next drug dose in a lower dose. Do the same thing the other way around as well, particularly if you have a cancer where response is easy to measure, such as leukemia. If you give a dose and there is no effect on the cancer measurable, and the patient had no adverse events, you can go up. That is adapting the dose to the patient, including intra-individual dose adaptation. That is also necessary to find the right dose and should be included in our dose escalation studies. And then there is probably the biggest of all. We want to give drugs that hopefully hurt the patient not, yet hurt the cancer. But all our dose finding is based on toxicity. That's only half of the picture. Here you have a graphic how it can happen. With different doses, you give higher doses, you have more effect. The blue line may be the line how the cancer dies. So with the dose of 6, you get a likelihood of 40% of the cancer shrinkage. And the reddish orange line is the one for toxicity. So with the same thing, you only have a probability of 15% to cause harm. The best drug is the drug that has those 2 lines far apart from each other. But the right dose is the dose that it works and doesn't hurt yet. And that is the gray line. We make our decisions only based on the toxicity line. It can't be right. Now, there is history behind it. And in the history, it was right. When I started all that treatment, there was no 3+3 design yet, and we did individual dosing every single time. And none of the drugs was approved. I'm a pediatric oncologist. So we gave the drugs based on what we thought was right from knowledge from other patients, but then we adjusted for the dose every single patient. The first diseases we learned to cure, at least in some patients, were acute lymphoblastic leukemia was the first one that was really successful. And I remember my teachers say when the first patients came, got protocol-driven therapy, and then didn't have much side effect. They called them, they sailed through the chemo. When that happened, they predicted that the patient's going to have a recurrence, and a recurrence of disease in those times was pretty much a death sentence. So when the patient was about to sail through chemo, they told us, give more. And so we did. Similar things are still true, but and they're still true for chemotherapy. They're not true anymore for our better drugs and with our far better diagnostics than just sail through. We have by pharmacodynamic, we can see the cancer if it shrinks or not, and know if the drug works. And our new drugs are far better than chemotherapy. They are very specific. We could start dosing based on effects as opposed to side effects. Well, I hope I have some listeners here among you that find this all exciting and will lead us to the next level of clinical drug designs for dose findings that will include all of those things will be much faster and better, and we will dose more patients right and cure more. So you made it through the entire video. Congratulations, I am honored. Let me close this by saying all these critiques and ideas of how things can be done better in the end really mean very little for today's practicality. 3+3 design is the leading way of doing phase 1 studies still, even though there are more intelligent methods available. And also, while I'm thanking you, you must have had something found in that video that you're still here. If you want to return the favor, please comment. Tell me what video to be done next. Thank you very much. Bye-bye.